Well, thank you and buongiorno, but that's the limit of my Italian, I'm afraid. So what I want to do today is to talk to you about bacteria, about epigenetics in bacteria, but having listened to some of the presentations already, I almost thought I should change my topic and talk to you about GMOs, genetically modified organisms, because GMOs are really very important in responding to climate change. If you look around the world, you see that in many of the developing countries, there are somewhere like 800 million people who go to bed hungry every night. If you go to bed hungry, this is not good for your health. If you're a child and go to bed hungry, this is especially bad for your health. But it's something we can fix. We can fix it by growing better crops, crops that have been edited properly, using CRISPR methods, using other methods, that will increase the yields of the crops, will increase the nutritional value of the crops, and will generally allow smallholder farmers to grow enough food to feed their families. And you might ask, why don't we do this? Well, I'm afraid the answer is because here in Europe, Greenpeace have taken over an agenda where they've made a lot of money by being opposed to GMOs. And I think this is something that has to stop. If we continue in this vein, we're killing people around the world. You know, recently, about a little over a year ago, something called golden rice was approved in the Philippines. Golden rice has a yellow color to the grain because it produces beta-carotene. This is the precursor of vitamin A. Golden rice is safe. It's been tested. It's been available for many, many years, except that Greenpeace have stopped it absolutely now in the Philippines, where it had just been approved, and then they filed a lawsuit and have stopped it again. You know, since this was developed, millions of children have died every year because they don't get enough vitamin A when they're growing up. Even today, hundreds of thousands of children die. And all of this because Europe has been opposed to GMOs, Greenpeace have been opposed, they go around the world spreading their message, and I find this unconscionable. I find this absolutely immoral. I don't know how these people sleep at night knowing they're killing children. So I could go on much longer. I'm happy to talk about it at a separate occasion, but today I'm going to talk to you about something else, which is something rather positive. And I'm going to talk to you about methylation in bacteria. Does this work? Yeah. So bacteria get a lot of bad publicity. Almost all of you in this room, when you were growing up, you thought that bacteria were bad because you hear about all the pathogens. And this was because since the very start of trying to understand bacteria, scientists, microbiologists, were looking at those bacteria that cause disease. And to some extent, this is understandable. We know that bacteria cause disease, you want to get rid of them. But as a result of this, if you just look down here from anthrax, gonorrhea, typhoid, plague, and so on, this was the predominant topic, and everybody started to think, well, all bacteria are bad. But in fact, it is totally untrue. The vast majority of all bacteria are good. If I kill the bacteria that live with us, and you'll see in a moment there are quite a lot of them, you will die. You can't live without them. They keep us alive. And we must always remember this, that bacteria are basically very good. If we're going to start implementing things, we need to implement things that will get rid of the bad ones. If we look at humans and bacteria, we have about 10 to the 13 human cells. we have 10 times as many bacteria. For every human cell, there are 10 times as many bacteria living with us. A lot of them live in our guts. They make sure that the food we consume every day is actually converted into useful metabolites for our bodies. This is crucial. If they don't do this, we die. This is why we don't want to kill them all. Now, human, one strain. Bacteria, there are more than 20,000 different types of bacteria that live with us. And all but a very few are really, really good for us. You look at the genetic information. There's more than twice as much genetic information in the bacteria that live with us as there are in the human genes that we have. Here are some representative microorganisms, some of the things that you possibly have heard of, you may not have heard of. Staphylococcus, most people have thought about Staphylococcus aureus. This can be a little dangerous, but Staphylococcus epidermidis lives in our skins and is really good for us. Similarly, in the mouth, many important bacteria. In the respiratory tract, in the gastrointestinal tract, in the urogenital tract. These are really a lot of different organisms. Some are a little dangerous, some are not. Here's an example of an unstifled sneeze. Okay, it's full of not just, I should say saliva or whatever you think, but not just water, but it's got a number of bacteria in it too. And so this is why if you have to sneeze, you should use a handkerchief or put your hand in front so we don't spread the bacteria around unnecessarily. Now I want to talk about microbial methylation, about what happens to the DNA in bacteria that really we know something about, but there's a huge amount we don't know. I think this is one of the wonders of being a biologist, is that there is so much we don't know. I always feel sorry for the physicists. You know, they know so much basic information, they know so much about how physics works. We as biologists, we know very little. Even those people who will stand up and tell you, oh, I know all about this, usually you can find plenty of things that they don't know about. Since I'm curious, since I love to make discoveries, I couldn't find a better subject to work on than biology. If there are any physicists in the audience, just give this a little thought. Now, DNA is very simple. It's made up of 4 bases, A, C, G, and T, but they get modified. They get a simple modification just putting a methyl group, a CH3 group, onto the bacteria. The 5 position of cytidine, 6 position of adenine, or the 4 position of cytosine. These methylations can have quite a big effect. They can be detected, for instance, by proteins. So a protein wants to come along and make a copy of a gene, make a messenger RNA corresponding to a gene. Sometimes the methyl group means that that gene can be transcribed. Sometimes it means it can't be transcribed. Sometimes it blocks it, it stops it completely. And so knowing what kinds of methylation are present really can be very helpful, and knowing where that methylation is can be even more helpful. Now in humans, of course, we also have some methylation, hence epigenetics in humans. In humans, it's almost all 5-methyl-C, the one on the top left here. A little bit of M6A can sometimes be found, but it's a little unclear whether that's genuine or not on the basis of what we know at the moment. But I'm not going to get into humans, I just want to tell you about bacteria. And what I say for bacteria also applies to archaea, which is the other kingdom of life. Now, why should one study these things? Well, I got involved because back in the early 1970s, I was interested in restriction enzymes. These are enzymes that cut DNA very specifically. They form the basis of cloning, allowing you to clone things. The biotech industry grew up because restriction enzymes offered you ways to manipulate DNA to study it, to find out what was going on. Now, these restriction enzymes cut DNA, and they're encoded by bacteria. And so why isn't the bacterial DNA cut up? And the answer is because the bacteria with a restriction enzyme also has a DNA methylase. That methylase recognizes exactly the same sequence as the restriction enzyme, and in so doing, it therefore defends the bacterial DNA against the action of the restriction enzyme. But then when a bacteriophage, a virus of bacteria, come and tries to infect, they usually do not have their DNA modified, and so the restriction enzyme can cut it and stop the infection, which is good. They're actually easily found by bioinformatics. They're good markers for RM systems. Several have been shown to have regulatory activity, and this is something I'm going to talk to much further. They also support epigenetic phenomena. That is, the DNA, when it divides, goes in, makes daughter cells, it starts off being methylated, and that can protect daughter cells. And they're a very good example of proteins that recognize DNA sequences. We're all interested in molecular biology about how proteins recognize DNA. How is it that they really find a sequence and do whatever they're going to do? But for me, the thing I love about methylases is that there are many more than appear necessary for their known biology. So I always thought for many years it was really just this restriction modification that they were important for. A couple of orphan methylases didn't have a restriction enzyme. We knew that they were responsible for a couple of things, but we never thought much about anything else. We now know that there are many, many more that are in bacteria, and we don't know what they do. Here's a question: what do they do? I'll give you a couple of answers. Let me first of all tell you a little side story. So when you win the Nobel Prize, you have to go to Stockholm and deliver a lecture. Most people will tell you about all the wonderful work they've done since they made their discovery. I discovered RNA splicing. I discovered how eukaryotes make messenger RNA by taking a very long transcript and then cutting and splicing it in much the way that you do when you make a movie to give a continuous messenger RNA. It turns out that I had stopped working on that probably about 6-7 years after I made the discovery, and instead I'd been working on methylases. And just 2 months before I had to go to Stockholm to give my Nobel lecture, we made this discovery. We discovered that when DNA methylases bind to DNA and want to transfer the methyl group, they take the base that they're going to modify and flip it 180 degrees right out of the helix. That's this yellow sign you can see up. The yellow is the base. And this led to all sorts of possibilities. It made you start to think about other ways in which DNA might be recognized, in which the ways other proteins might be recognizing DNA and could do it. And we now know that almost everything that we know of that modifies the bases in DNA uses this mechanism in order to do so. So here I am, I'm in Stockholm. I've got the Nobel Committee sitting in front of me. I've got loads of scientists there. So I told them about this discovery. You know, here's the opportunity for yet another Nobel Prize, perhaps. I don't know. No. If it came, it would be because I work with a crystallographer, and he would be the person to get it, I hope, anyway. So I just want to tell you a little bit about restriction modification systems, just so you get the idea here. And there are 3 basic kinds. The type at the top, the type 1, they are 3 genes. One methylates DNA, shown in blue, that's a methylase. The yellow gene is a specificity subunit, and it recognizes the specific DNA sequence that is going to be methylated. The restriction enzyme is the reddish-orange colored gene, and it cuts the DNA. And these systems operate by methylation, requires the S subunit for specificity and the M subunit to do the modification. So the S subunit recognizes the sequence, and if it's going to cut, you need all three subunits to come together, but the cutting is random. The cutting goes all over the place. It's not specific. The methylation is specific, but not the current. And so that makes it very hard to determine recognition sequences. Now, the type 2 enzymes are much simpler. They are actually two completely independent enzymes. Methylase, shown in blue again, that recognizes the specific sequence, and the orange gene, the R gene, this recognizes exactly the same sequence and cuts it. And this means it's easy to determine their recognition sequence because you just take a sequence piece of DNA, you know what the sequence is, you cut it, get a bunch of fragments, find out what's at the end of each fragment, that's the recognition sequence. So that was easy. Methylation, though, was very difficult. We didn't have a good way of doing it until a few years ago. Type 3 enzymes are similar to the type 2, R gene and M gene. The R gene was always rather tricky to figure out where it was cutting because it didn't always cut specifically in the way that one wanted, and it was often strand specific also. And that turned out to be hard. And so it turned out it was quite hard to get the methylation sequences to find out what was being methylated, which is what I wanted to know. I want to know what these things are doing and how they do it. So along came a system that I don't want to go into the detail of because it will take too much time, but along came a system from a company called Pacific Biosciences, and they came up with a new way of sequencing DNA, which they did by copying the DNA using a specific polymerase. And the polymerase they used and the way in which they did the sequencing meant that as the polymerase was copying the DNA, if it came to a methylated base that it was trying to copy, it slowed down. And they were able to measure the speed at which each base was added to the growing DNA chain. And in this way, they were able to find out where the methyl groups were. And so all of a sudden, we had this tool, very simple. We just had to use PacBio sequencing, and we could get the methylation sites. It took a little software to work out, where it was stopping, what the sequence was, and so on. But that was all relatively straightforward and was all done. And so we started sequencing a lot of bacterial genomes this way, and in particular, just not to get the sequence, because many had been sequenced by old methods, but to find out where all the methyl groups was. This is an example of an E. coli strain that caused hemolytic uremic syndrome in patients in Germany. It was quite widespread. And we sequenced it, and the genes that we found are all listed here, methylases and then the companion S and R genes in the case of the type 1 systems. But I just want to draw your attention to two of these methylases here. The first two shown are M-ECHO-G2 and M-ECHO-G1. And they have the property that they methylate every single A residue. So whatever your sequence is, CGTA and so on, every single A was methylated by these enzymes. And it turned out these hadn't been seen before. This was the first time that this was seen. And you can see what it looks like, very easy. And it turns out that this is very useful if you want to take DNA and put it into a strain that you know has a restriction system. You can protect it by methylating every single A residue using these enzymes. And that turns out to be quite interesting. There are other uses for it too. Our company now sells it. Now there's another very interesting enzyme that was present called M.ECHO-G9, shown again in yellow at the bottom here. And this had the property that it just methylated one strand of the DNA, and it was absolutely strand specific. And if you, there was another one found in an 𝗵𝗂𝗶𝗰𝗀 species that was very similar. But it turns out that if you do the following experiment, it's encoded on a plasmid. if you flip the strands of the plasmid, which you can do just by altering the DNA replication origin, depending upon which way the origin is pointing, that points to the strand that gets methylated. So it's clearly related to DNA replication, and it's related to when the plasmid wants to move from one strain to another. This is the way it protects itself against restriction if that is in the other strain. Now, another big project that I've been involved in is with Helicobacter pylori. Many of you may have heard of H. pylori. In fact, it was for a long time thought just to cause ulcers. We now know it actually can cause cancer. It's one of the few bacteria that we know for sure that can cause cancer. And I've been involved in a big project with the National Cancer Institute in the USA to look at really more than 1,000 Helicobacter pylori strains that have come out of cancer patients around the world, trying to understand what it is about the strains that cause cancer that are different from the ones that don't. They have a lot of different methylation systems. In fact, Helicobacter pylori has more methylases than any other bacterium we know at the moment, absolutely loaded with them. I've shown here various motifs. I would just draw your attention to the unique motifs. So within about 1,200, a little over 1,000, about 1,050 strains, type 1 enzymes, these 3-gene enzymes, there are more than 350 specific sequences that are recognized. This is typical of what happens if you've got a system that is protecting against phage. Okay? They vary. They keep varying because otherwise the phage figures out how to overcome it. It becomes methylated at these sites and then it goes. And so bacteria have to keep changing specificity. The type 2 enzymes, which we always used to think were really important for restricting incoming DNA, among 1,050 Helicobacter strains, there are just 55 different specificities. Type 2G is a special kind of type 2, 15 of those. Type 3 are 21. But the important thing is the difference in numbers between 370 for the type 1s and the others. And so it becomes a little bit curious. These are the sequences recognized by these type 2 enzymes, or at least some of them. And most Helicobacter pylori strains have 8, 10, 12 of these present in their genomes. The one that recognizes GC is present in every Helicobacter strain. And this suggests to me that these things are doing something other than protecting against phages. It's the type 1 enzymes, these 350 odd. These are the ones that are protecting against phages. And so what are these doing? We don't know. The bottom line is we don't know. And I have to tell you about a story. I was in a hotel room in Japan giving a lecture to a group in Novosibirsk in Russia. And I was talking about restriction enzymes and all of this stuff. And afterwards, someone in the audience said to me, well, could it be that the restriction enzymes are not there to defend phage, just happens to be useful, but rather they're there to make sure that the methylase keeps its specificity? Because if the methylase were to lose its specificity and start methylating some other sequence, restriction enzyme would kill that bacterium. I'd never thought about it. And I've been trying now for more than 5, 6 years to locate this person in the audience in Novosibirsk who came up with this suggestion, because I thought it was a fantastic one. And I have a feeling it's probably correct. But I can't. I contacted the organizer of the meeting. I contacted people at the meeting. None of them knew who this person was. So if ever you come across someone who tells you about this great idea, let me know. Because I really want to give credit to this person. I thought it was such a good idea. So what have we learned so far about this? Well, we found that there are many solitary methylases in bacteria and archaea. And many are probably involved in epigenetic phenomenon, but we don't know how many of them there are. There's much more variability in recognition sequences than we ever expected. The type 2 methylases and the type 2 restriction enzymes, these are two separate components, two separate enzymes. They evolve separately, but they often have exactly the same specificity. Sometimes the methylase has other sites, a few extra sites that it methylases, but it doesn't matter from the point of view of the restriction enzyme. And there are many new non-specific methylases that have been discovered on prophages, that is, virus genomes that have incorporated into the bacterial genome. Now, I want to really tell you a little bit about something that I'm particularly excited about, and that's these orphan DNA methyltransferases, the ones that do not have a restriction enzyme associated with them. There are two that are really well known, 𝗘𝗃𝗃. This is an enzyme recognized as GATC, and it's used in mismatch repair, absolutely crucial for mismatch repair. Another one in E. coli, recognized as CCWGG, has been known for 40-odd years now. We don't know what it does. We have no idea what this does. E. coli is probably the most studied microorganism out there. It's got an enzyme here. We don't know what it does. ECHO-K2. Hey, this is another enzyme in E. coli, recognized as ATGCAT. Normally, when E. coli grows in you in the lab, it's not expressed. There's no known biological role for it. And the only reason that we know what it recognizes is that you can clone the gene and then look at it outside of E. coli, and so we can figure out what it is. But until a few years ago, no one had ever seen it expressed in E. coli. And I was in a meeting in Lindau. They have a meeting every year of Nobel laureates, and they have loads of students there. And I was there, and I happened to be talking to a student who was talking about the fact that when E. coli grows as a biofilm, that is, it grows just on a film rather than growing in culture, there were 2 or 3 genes it expresses that are only expressed in the biofilm. And so I was talking to this young lady, and I said, well, you know, could you send me an extract from one of these biofilms where you found, and lo and behold, this methylase was expressed. The only time it's been seen to be expressed in E. coli, what it's doing there, why it does, we don't know. But these are the questions I love. I love things where I don't know what's going on. Now I've got a purpose in my life to find out what's going on if I can. Another methylase is CCRM1 in Colobacter. This one is controlling cell cycle progression. Quite interesting. We don't know the full details of how it does it, but we know enough to know that this is absolutely crucial for cell division. The enzyme, the methylase I'm most excited about at the moment is this one, M.CDI361. It's a DNA methylase found in Clostridium difficile. Clostridium difficile is this organism that if you go to hospital and spend any length of time there, you'll probably get a C. diff infection. And C. diff is really difficult to control. Most of the strains are already antibiotic resistant, but typically when an antibiotic comes along, this strain starts forming spores. That is, just little eggs, if you like, that don't grow. And it sits there, waits until the antibiotic goes away, and then it starts growing again. And it's been unbelievably difficult to control. And what happens if you pick it up when you're in hospital, they can't do anything about it, and so they send you home. And that would be okay, except the symptoms of C. difficile infection are uncontrollable diarrhea. Right? Now, you can just imagine if you have diarrhea anytime, at any place, you've no idea when, all of a sudden it just happens, and so patients go home, never leave their home. They don't want to get on a bus, they don't want to get in a car and suddenly have diarrhea. Now, fortunately, there is one thing. This is the organism, Clostridium difficile. One of the things about it is that there is a solution. There is a way that you can cure it. And that's shown on this next slide. 𝗬𝗂𝗃𝗃𝗃. It's been found that if you take extracts from fecal material from a healthy patient, then that fecal material actually will kill C. difficile. We don't know how it does it. We don't know what's going on. There's obviously something there that does it. We don't know what it is. They have found a way of administering fecal material. So they have collections and so on. You know, of course, I put here this artificial pill, which tells you what a fecal transplant looks like. They don't actually look like this. This was just done as a joke by the lady, Elizabeth Homan, who discovered the pills and figured out how to do it. But you can imagine, but fecal transplants, you can't get them everywhere. They are available in some places. You have to test the feces first to make sure there's nothing there that you don't want and so on. So it's not so straightforward. So I want to stop at that point because this is just one example of an orphan methyltransferase, because if it turns out you block the expression of that methyltransferase, C. difficile cannot sporulate anymore. You've solved the problem. You can solve it. And there are now 2 drugs in clinical trials based upon this discovery that we hope are going to provide a cure for C. difficile, just by stop it sporulating, and then regular antibiotics will help you. As you can see, there are other large number of people who I've worked with over the years. A huge number send me PacBio data. They send me the data, and I work with them. I collaborate with them. I love collaborating. And so that's been very important. And I always like to finish lectures these days by talking about some of the problems that women have faced in science. I really want to recommend these books to everybody. One called Invisible Women, written by a British author, and looking at the ways in which society has a systematic bias against women. You know, doctors, when they were doing clinical trials, who do they choose as their subjects? White men. Women, well, occasionally, but usually white men. Blacks are discriminated against as a result of this. Women are discriminated against as a result of this. The lady who won the Nobel Prize last year, 𝗘𝗃𝗃𝗃𝗃, if you read this book, it's an autobiography, came out just before Christmas. It is one of the most incredible books I've read. What she went through, just being treated badly from day one, is absolutely incredible. And I think it's a lesson to us all that we should read these books and find out the ways in which we men have mistreated women over the years. And I'm no exception. I've done it. We don't always know that we're doing it, but we do it. This book by 𝗘𝗃𝗃𝗃𝗃, The Exceptions, is about a lady called Nancy Hopkins, who was a professor at MIT. When she first arrived as a professor, they wouldn't give her a technician. They gave her lab space that was a half of that of any man. And she slowly figured out what was going on. And there were at the time, I think, 11 other ladies who worked as professors at MIT, 150 men. And she went around and measured the lab space that the women had as compared to the men. If it was half, they thought they were lucky. They wouldn't give Nancy a secretary, right? Women don't need secretaries. And so she rounded up the women. They documented everything and went to the president of MIT, and he listened and changed the rules. Very, very good book, well worth reading. And then this picture on the right, this is a little documentary called Picture a Scientist, again, about Nancy Hopkins, but about other women who'd been mistreated in science. And I think it really is important. This is something that men, for sure, should read, just so that you realize, so that we realize how we do mistreat women from time to time. We don't mean to. We're not doing it deliberately, but we inadvertently do it. For instance, if you treat a woman as a man when it comes to the job you expect her to do, it doesn't work. Women have children. They need maternity leaves. They need time in order to be a woman. And we have to recognize that they cannot and should not be asked to behave as men. But we should take note of the fact that usually they're a lot more empathic than men. They feel a lot more about the people about them. They make very good mentors. We should recognize this. And so I say all, please read these books, see this documentary. They're excellent. And at the bottom is a final note. This is a website about GMOs. We have 171 Nobel Prize winners all say that Europe should change its attitude towards GMOs. And they're all listed on this site. You can log on, you can join us, and there's a lot of stuff about the GMO campaign on there. And with that, I thank you all for your attention.